Welcome to this video on protein import into the nucleus. In this video, I will explain how proteins are transported from the cell cytoplasm into the nucleus. The focus will be on the mechanism of nuclear protein import, highlighting the critical role of Ran-GTP system in regulating directionality and cargo release. I will explain the features of nuclear localization signal sequence, importin receptors, energy requirement or Ran-GTP system, and finally the nuclear pore complex. To understand the mechanism by which the nucleus imports proteins from the cell cytoplasm, I will review the essential features of these requirements. The requirements are as follows. First one is nuclear pore or channel. Second is cargo protein with nuclear localization sequence. Third is receptor or importin. And fourth is Ran-GTP. The nuclear pore complex is quite complex and it is made up of thousands of polypeptides. On the cytoplasmic side, there is a ring which is attached to the filaments. These filaments guide the cargo to pass through the channel. The another ring is on the nuclear side. This is known as nuclear ring. This is also connected to the filaments, making a structure known as nuclear basket. There is a central ring which is the main component of the channel. This forms the channel made up of several different type of proteins. The interior of the channel is lined with the special protein. They are known as FG repeat proteins. FG here is phenylalanine glycine repeat. These proteins create selective barrier to help the molecule to pass through this channel. So basically you need a nuclear localization signal or sequence to be present in the cargo protein at their N terminus. The nature of the NLS is positively charged because of the presence of the basic amino acids such as lysine and arginine. They may be having one stretch of these amino acid, positively charged amino acid, and such NLS is known as monopartite. In some cases, you may have two stretches of positively charged amino acid separated by 10 different amino acids. This type of NLS is known as bipartite NLS or signal sequence. Now these signal sequence is very important for targeting the protein to the nucleus. If there is any mutation on these sites, the protein will not reach nucleus. There will be deficiency and therefore producing many kind of pathologies or diseases. So the next requirement is the receptor or importin. The nuclear proteins carrying NLS are selected by this receptor, which is also called as importin. The receptor does three things. First, it selects. Second, it stabilizes. And third, it transports the cargo protein or the receptor cargo complex through the nuclear channel. The receptor is made up of essentially two subunits, alpha and beta subunit. Alpha subunit is responsible for selection of the cargo having NLS in their N-terminus region. Beta subunit on the other hand is responsible to import or to transport this cargo and receptor complex through nuclear channel. Couple of points about Ran-GTP. It is a monomeric G protein, also known as RAS-associated nuclear protein. This is in higher concentration in nucleus and less in the cytoplasm, and this is due to presence of the guanine exchange release factor in the nucleus. Ran-GTP helps in recycling the importin receptor into the cytoplasm. In addition, it also helps to release cargo in the nucleus. So let us discuss now import mechanism. This is the basically nuclear membrane, outer nuclear membrane, inner nuclear membrane. As I mentioned earlier, that at points these both membranes they fuse together, making pores, one here, one here, and one here. So this is cytoplasmic site, and this is the nuclear compartment. The protein has to be imported from cytoplasm into the nuclear compartment. So the first step is the selection of the cargo, which is based on the presence of NLS or localization sequence at N-terminus by the receptor, which is also called as importin. So it will bind, and this whole complex in step 2 is transported through the pore into the nuclear compartment. So for this step there is no need for any energy, so it is energy independent step. The second step is completed. Therefore now the third step is release of cargo, which is liberated by Ran-GTP. It has high affinity for receptor, thereby releasing the cargo protein. Cargo protein is fully folded. There is no need for further folding of this protein. It is functional. The second point is that the localization sequence is retained in the protein. It is not deleted like any other protein. So Ran-GTP form complex with the receptor. So this complex R and Ran-GTP in step 4 moved through the pore into the cytosol, and while it is moving there is a protein called gap which activate Ran-GTPase activity. As a result of that, importin or the receptor is liberated, producing Ran-GDP, which in step 5 is transported back to the nucleus by the action of NTF2 or nuclear transport factor 2, which in the nuclear compartment is converted into Ran-GTP with the help of GEF or guanine exchange factor. So in the nucleus you have high concentration of Ran-GTP, whereas low concentration in the cytoplasm, and this concentration gradient is maintained and is important for the normal functioning of import mechanism of nucleus. Thank you for watching this video. I request you to like it, share it and subscribe it. And until next time, you take care. Bye bye.